Okay, so we'll start today's session. I'll share my screen. Can someone unmute and let me know if I'm audible and if my screen is visible?
Yes.
Okay, thank you. Okay, so we'll start. I can admit people as and when they come in. So, good evening, everyone. Welcome to yet another session of this course, Applied Environmental Microbiology. My name is Aishi, and I'm a PMRF research scholar at the Department of Chemistry, IIT Bombay, and I'm a PMRF TA for this NPTEL course, Applied Environmental Microbiology. So what we do in these live sessions is that we discuss some of the sample questions, maybe of this year's assignments or last year assignments, and we'll discuss them, and we hope the session turns out to be as interactive as possible. And we hope that this problem solving session will help you solve questions in the upcoming assignments that you have to submit within the due date. So, welcome to another session. So, before I start the session, I would like to mention that during the last session, we were talking about some of the technical glitches on the website, like some of the answers, some of the topics that have been already taught in the course, but uh, the answers you were right, like you were giving right, correct answers on the portal, but still the portal was showing wrong answers to be correct. So I have informed the NPTEL staff regarding this issue, and they have forwarded my query, my concern to the required people from IIT Roorkee. So hopefully the technical glitch or whatever the issue is, it will get solved as soon as possible. So moving on to the first question for the day. So, detection of organic matter in drinking water is fairly difficult due to their complex structure. So, instead, an estimation is done using carbon or uranium content. What do you think? Is it true or false? Anyone? Do you think the statement is true or false?
False.
Okay, and why? Why do you think it's false? You have any ideas in mind? Anybody else? Yes, so this the statement is not exactly factually correct. This is false, and that is because there are different types of organics in water. So, as you can see in the slide also, the types of organic matter in water can be very diverse and may be present in suspension or colloidal or true solution or whatever. And organics in water can come from various sources like vegetable matter, runoff from fields, domestic and industrial waste. And one, there are some criteria, some classifications or categories on how this can be segregated, and these topics are quite like very popular in microbiology, such as the biochemical oxygen demand. I think I have talked about this term 2 to 3 times in the live sessions. So it's a very popular term in microbiology. Biochemical oxygen demand is basically an indirect measure of the biodegradable organic compounds in water. So if there are microbes present, if they are taking in, if they are using oxygen to degrade any organic matter in water, then the biochemical oxygen demand will increase. So like the a very high value of BOD means the sample of water is very polluted, like it's very dirty, it has a lot of microbes in it. So this thing is a measure of the kind of microbes that we find in various samples. Then we have nutrients. Nutrients are obviously chemical elements or compounds, and they're essential for growth of any living being, a plant and animal. And nutrient parameters include ammonia, organic nitrogen, gel and nitrogen, nitrate nitrogen, and total phosphorus. Then we move on to organic carbon, and most organic carbon in water occurs as partly degraded plant and animal materials, and some of these are resistant to microbial degradation. Then we have oil and grease. Oil and grease constitutes one of the most common parameters for quantifying organics from human sources, and these tend to be from illegal dumping into the storm sewers, motorboats, oil spills, and discharge from oil production platforms in the sea. So what happens is that how do we measure this organic matter in water? So organics in water can be tested by the total organic content, which is known as TOC, or the sum of particulate and dissolved organic carbon. Dissolved organic carbon is TOC, also known as the humic substances or refractory organic substances. And quantifying organic matter remains a challenge. It does remain a challenge due to its complex nature and variability from source to source. Element experts can detect natural organic matter through many advanced spectroscopic, chromatographic, and mass spectrometry techniques. For routine, non selective monitoring, the experts basically mostly typically they rely on quantifying organic carbon content, that is, measuring total or dissolved organic carbon, or estimating the hydrophobicity using specific UV absorbance, and identifying oxidation potential using photoelectrochemical chemical oxygen demand basically. Now we know that public water is regulated, but still sometimes they have the capacity to get contaminated. So there is this US Environmental Protection Agency, or the EPA, which has regulated drinking water quality in public water systems, and they basically set limits for germs and chemicals in water. However, still sometimes unsafe levels of harmful germs and chemicals can contaminate public drinking water, and the germs and contaminants can get in water at its source. For example, groundwater, or water that is coming from lakes or rivers, or while water is traveling through the distribution system also, after the water treatment plant has already removed germs and chemicals from the source water. So this EPA, they require water utilities to test for and address many germs and chemicals in their drinking water systems. So water utilities are required to tell customers about testing for contaminants and whether they found any unsafe levels of chemicals or germs in the water. And customers are then notified through notices of violations and drinking water advisories. And water utilities must also provide their customers with an annual drinking water quality report called the Consumer Confidence Report. And we know that not everyone is equally prone to any kind of disease, or any kind of not equally susceptible to any kind of germ. So some people are more likely to get sick from germs and chemicals in water, such as infants, young children, people who are pregnant, older adults, people who have really weakened immune systems, some comorbidities they have, such as people living with HIV, getting chemotherapy treatment, or taking transplant medications. All these people have much higher chance of getting affected by contaminated water. Now, how does the water get contaminated? So, germs and chemicals can get in drinking water, as I said, at the source or at the distribution system when the things are getting distributed. And harmful germs and chemicals can also include fertilizers, pesticides, or any other chemical that has been applied to land near the water. Then, concentrated feeding operations like large industrial animal farms, then manufacturing operations, sewer overflows, storm water, wildlife, rocks and soil that naturally have chemicals and minerals such as arsenic, radon, and uranium, then cracks in water pipes or other problems in the distribution system basically. So, what are the prominent steps for this water treatment? Coagulation. Coagulation is often the first step in the water treatment, and what happens during coagulation is that chemicals with a positive charge are added to the water. So, the positive charge neutralizes the negative charge of dirt and other dissolved particles in the water, and when this occurs, the particles bind with the chemicals to form slightly larger particles. And common chemicals used in this step include specific types of salts, aluminum, or iron. Then we have flocculation. Flocculation follows the coagulation step, and in flocculation is basically the gentle mixing of the water to form larger, heavier particles called flocks. and often water treatment plants will add additional chemicals during this step to help the flocks form. Then we have sedimentation. Sedimentation is one of the steps the water treatment plants use to separate out solids from the water. So during sedimentation, flocks settle to the bottom of the water because they are heavier than water. Then we have filtration. So once the flocks have settled to the bottom of the water, the clear water on top is filtered to separate additional solids from the water. So during filtration, the clear water passes through filters that have different pore sizes and are made of different materials such as sand, charcoal or gravel. And these filters remove dissolved particles and germs such as dust, chemicals, parasites, bacteria and viruses. And activated carbon filters also remove any bad odors. Then water treatment plants can use a process called ultrafiltration in addition to or instead of traditional filtration. And during this ultrafiltration, the water goes through a filter membrane with very small pores. And this filter only lets through water and other small molecules such as salts and tiny charged molecules. Then reverse osmosis is another filtration method that basically removes additional particles from water. And water treatment plants often use reverse osmosis when treating recycled water or reused water or salt water for drinking. Then there is a step called disinfection. So after the water has been filtered, water treatment plants may add one or more chemical disinfectants such as chlorine, chloramine, or chlorine dioxide to kill any remaining parasites, bacteria, or viruses. And to help keep water safe as it travels to homes and businesses, water treatment plants will make sure that the water has low levels of the chemical disinfectant when it leaves the treatment plant. And this remaining disinfectant kills germs living in the pipes between the water treatment plant and your tap. So in addition to or instead of adding chlorine, chloramine or chlorine dioxide, water treatment plants also infect water using ultraviolet UV light. Like UV light and ozone work well to disinfect water in the treatment plant, but these disinfection methods do not really continue killing germs as water travels through the pipes between the treatment plant and your tap. So you are exposing your water to the UV and ozone at a particular time point, but then you'll have to stop. Like you cannot, the entire transport from the distribution pipe to the houses, it is not monitored with the help of UV or ozone. So even if you disinfect your water using these techniques, then also there's a huge path that the water has to travel, and at that point it might get contaminated. Tertiary treatment is the next sorry wastewater treatment process after secondary treatment. So what happens in this step is that this step removes stubborn contaminants that secondary treatment was not able to clean up, and wastewater effluent becomes even cleaner in this step process through the use of stronger and more advanced treatment systems. So tertiary treatment technologies can be the extensions of conventional secondary biological treatment to further stabilize the oxygen demanding substances in the wastewater or to remove nitrogen and phosphorus. So these are the types of organics in water basically, and why the statement was wrong is because there's no estimation regarding including carbon and uranium required, because it's pretty even though challenging, it's pretty common to use BOD and all this TOC and all these values to detect the organic matter in water. And as I said, there is also a tertiary treatment facility which can be an extension of conventional secondary biological treatment to further stabilize the oxygen demanding substances in the wastewater or to remove nitrogen and phosphorus. And tertiary treatment may also involve physical chemical separation techniques such as carbon adsorption, flocculation, precipitation, membranes for advanced filtration, ion exchange, dechlorination, reverse osmosis, etc. So advanced sewage treatment generally they involve three main steps: primary, secondary and tertiary, but may also include intermediate stages and final polishing processes. And the purpose of tertiary treatment is to basically provide a final treatment stage to further improve the effluent quality before it is discharged to the receiving water body. And more than one tertiary treatment process may be used at this treatment plant. So these are some of the steps how drinking water is treated. And even though all these steps have something in commonality, like they help disinfect the water, most of these have some kind of cons as well. So that is how the entire process works, and basically you have to weigh in the pros and cons before you decide on a particular technique. So moving into the next question, 100 ml wastewater sample is diluted to 500 ml in a BOD bottle using distilled water. After 5 days of incubation at 20 degrees Celsius, The initial and the final dissolved oxygen concentrations were 9 mg per liter, 3 mg per liter, respectively. What is the BOD of the wastewater? Anybody? You can try the sum in your copy. If you have a copy with you right now, you can just try out the sum. Let me know the answer. Anybody? You can just try it out. Anybody would like to try? It's a very simple calculation.
30 mg per liter.
Yeah. Okay. How did you do it? Yes, you're correct. Okay, I'll explain. So it is 30 mg per liter. But before that, let me tell you what is BOD5. What does BOD5 mean? So biochemical oxygen demand is BOD, as I discussed previously. It refers to the amount of dissolved oxygen needed by microbes to break down organic material present in a given water sample at a certain temperature over a specific time period. So BOD is measured in a water sample during 5 days of incubation at 20 degrees Celsius generally. So that is why it is known as BOD5. This term is because 5 days of incubation are involved. So basically it gives a measure of the amount of organic matter which can be decomposed by aquatic organisms. It also indirectly represents the degree of organic pollution of the water. So this is how the calculation will be. The initial dissolved oxygen was 9 mg per ml. 9 mg per liter. So that's the initial DO. The final dissolved oxygen was 3 mg per liter according to the question. So these are the values. The dilution factor was 5. As you can see, there are 100 ml wastewater and diluted to 500 ml. So the dilution factor was 5. The dilution factor 1 is to 5 means that 100 ml is diluted to 500 ml. Therefore, BOD5 is a very basic formula. BOD5 will be DO, dissolved oxygen initial, minus dissolved oxygen final into the dilution factor. So that is how 30 mg per liter, we get the value of 30 mg per liter. Is there anybody who has a different answer or who doesn't agree with this? Anybody? It's a very basic calculation. Okay. So great. So we can move on to the next question. So the tertiary treatment in wastewater treatment involves. I was already talking about it in brief in the previous question. Anybody?
Last option.
B and C. Okay. Anybody else? Yeah. So it is BNC, that is removal of nutrients and removal of persistent contaminants. So what is tertiary treatment of wastewater basically? It's the treatment after secondary treatment has already been done. So basically this step removes all the stubborn contaminants that secondary treatment was not able to clean up, and wastewater effluent becomes even cleaner in this treatment process through the use of stronger and more advanced treatment systems. Tertiary treatment technologies can be the extensions of conventional secondary biological treatment to further stabilize oxygen demanding substances in the wastewater or to remove nitrogen and phosphorus. Tertiary treatment may also involve physical chemical separation techniques such as carbon absorption, flocculation, precipitation, membranes for advanced filtration, ion exchange, dechlorination, reverse osmosis, and all this. So, how does this work basically, tertiary treatment? So, advanced sewage treatment generally involves three main steps called primary, secondary, and tertiary treatment, but may also include intermediate stages and final polishing processes. So, the purpose of tertiary treatment is to provide a final treatment stage to further improve the effluent quality before it is discharged to the receiving water body or reused. So, more than one tertiary treatment process may be used at any treatment plant. If disinfection is practiced, it is always the final process. It is also called effluent polishing. Now, tertiary treatment may also include basically may include biological nutrient removal. It can alternatively, sometimes it is also classified as secondary treatment, but it also involves disinfection and removal of micropollutants such as environmental persistent pharmaceutical pollutants. Tertiary treatment is also sometimes defined as anything more than primary and secondary treatment in order to allow discharge into a highly sensitive or fragile ecosystem such as estuaries, low flow rivers, or coral reefs. So, treated water is basically sometimes disinfected chemically or physically by lagoons or microfiltration prior to discharge into a stream, river, bay, lagoon, wetland, or it can be used for the irrigation of a golf course, greenway, or park. And if it is sufficiently clean, it can also be used for groundwater recharge or agricultural process. Now, salt filtration removes much of the residual suspended matter, and filtration over activated carbon, also called carbon adsorption, removes residual toxins. And then there's the microfiltration thing. So microfiltration or synthetic membranes are used in membrane bioreactors and can also remove pathogens. So settlement and further biological improvement of treated sewage may be achieved through storage in large human-made ponds and lagoons. And these lagoons are highly aerobic and colonization by native microphytes, especially breed, is often encouraged. So this is the final stage of the multi-stage wastewater treatment cleaning process, and it removes inorganic compounds, bacteria, viruses, parasites. So removing these harmful substances makes this treated water safe to reuse, recycle, or release into the environment. And how do you prepare wastewater for this tertiary sewage treatment? So basically, as I already said before in some of the previous classes, primary treatment of wastewater involves filtering out large solid contaminants. Secondary treatment of wastewater then purifies the wastewater through biofiltration, aeration, and oxidation. These are all the processes that help to remove sediment from the water. So the municipal water treatment solutions at the any kind of segregation will help you execute the three stages of wastewater treatment, and through this responsible three-stage water treatment, they'll protect both the people and the natural environment from the harmful effects of untreated water. So this last step will have final filtration of the treated effluent. When needed, it can also sometimes involve using alum to remove phosphorus particles from the water. Alum also causes any solids that were not removed by primary and secondary wastewater treatment to group so that they can be removed by filters. And when necessary, the filters can be filters are backwashed to remove the buildup of floc, which allows the filters to continue operating effectively. After this, there will be a disinfection process where the addition of chlorine is done to the final effluent before discharge, and this process injects chlorine into the headworks of a serpentine effluent detection chamber. So chlorination in wastewater treatment kills bacteria and viruses and eliminates parasites such as Giardia and Cryptosporidium, which can cause very serious illnesses. In summary, this process disinfects water so that it is safe to reuse or recycle. Then there is dechlorination. So the final stage of the tertiary wastewater treatment process involves removing the chlorine that was used to disinfect the water in the first place. So this step is very important because chlorine is very harmful to aquatic life. Chlorine also reduces biological water quality when it is present in high concentrations. To remove the chlorine, a compound called sodium bisulfite is added to the water. Chlorine ions in the water react with this chemical and are removed. Now, once the chlorine concentration has been reduced to a safe level, treated water is now considered clean enough to be safely released into the environment. So that is how the entire disinfection and the treatment of tertiary treatment thing works out. So yes, you were correct. The right answer is B and C, which is removal of nutrients and removal of persistent contaminants. Okay. Moving on to the next question. Which of the following organisms are involved in biological phosphorus removal process? Anybody? It's quite self-explanatory from the option only, but still, anybody would like to try?
Option 1.
Polyphosphate accumulating organisms and glycogen accumulating organisms. Anybody else? Anybody? So yeah, she is correct. It is option 1. It is the polyphosphate accumulating organisms and glycogen accumulating organisms. So recent research on the process of biological phosphorus removal in lab scale treatment systems, it has indicated that the development of an actively polyphosphate accumulating bacterial community after the introduction of an anaerobic period may take at least 4 months. Up to 80% of all aerobic bacteria isolated from these communities are able to accumulate polyphosphate. And polyphosphate synthesized by the bacterial communities of lab scale treatment systems is probably mainly low polymeric, not exceeding 20 P residues, and this poly P is rapidly degraded during the anaerobic period. The enzymatic hydrolysis of poly P under anaerobic conditions is accompanied by polyhydrobutyrate formation from exogenous acetate. Reducing equivalents are provided by the degradation of the carbohydrates, and also nitric oxide inhibits the release of phosphate under anaerobic conditions in various FND sludges. So bacteria belonging to the genus Acantobacter occur in a wide variety of activated sludges in which enhanced biological phosphate removal is observed. So I think as Acantobacter johnsoni 210A was studied in detail with respect to the elemental composition of the polyphosphate granules, the enzymatic synthesis and degradation of polyphosphate, the regulation of polyphosphate metabolism, and the transport of phosphate. So this particular bacteria reflects activated sludge in a number of ways as far as polyphosphate metabolism is concerned, but its polyphosphate is highly polymeric and the phosphate efflux rate under anaerobic conditions is relatively low and not increased by any kind of exogenous acetate. So in addition to Acantobacter, other polyphosphate accumulating microorganisms may be involved in the biological phosphorus removal. So the isolation of polyphosphate accumulating denitrifying bacteria may well have interesting applications for a new process design in wastewater treatment systems. Further studies on the enzymes involved in this poly P biosynthesis and on the uptake and the efflux systems of phosphate, potassium, magnesium, and lower fatty acids in pure cultures will enlarge our insight basically into the genetics of the metabolism of polyphosphate. In addition, the regulation of the metabolism of polyphosphate accumulating organisms needs to be studied in more detail. And one of the most serious water quality problems of the last decade is the eutrophication of lakes and slowly flowing rivers. So this results in the overabundant growth of algae and macrophytes, and both phosphate and nitrogen are often the limiting factors for biological growth in natural waters. However, cyanobacteria are capable of fixing molecular nitrogen, thus eliminating the requirement for ammonia or nitrate for their growth. In addition, they obtain metabolic energy by photosynthesis and use CO2 as a source of carbon. And in practice, growth prevention only needs a lowering of the phosphate availability. Experiments with large water reservoirs have shown that no eutrophication occurs when the phosphate concentration is reduced to 8 to 10 mg per liter, 8 to 10 xgl. Like even the nitrogen concentration amounts to 4 to 5 mg basically. So phosphorus removal process, unlike removal of nitrogen, this phosphorus removal can only be carried out by the incorporation of phosphate into a solid phase, which can be separated from the water by setting flotation or filtration. And the process involved in the phosphorus removal have been excellently reviewed by a paper authored by Megank and Fobb. They basically include fluidized bed crystallization, ion exchange, electrochemical phosphorus removal, treatment with algae, deep bed filtration, absorption, precipitation by iron oxidizing bacteria, and chemical precipitation. So it has been said that in the Netherlands, a conventional treatment plant removes about 40% of the 15 mg phosphate present in the domestic wastewaters, and the removal is due to the growth of the biomass, which contains 50 to 20 mg 50 to 20 mg phosphate dry weight. So the remainder of the phosphate in the wastewater is usually removed by chemical precipitation with calcium, iron and aluminum salts, resulting in a final left rate concentration of about 1 mg. So chemical precipitation still has several disadvantages. That is increasing the cost due to the use of considerable amounts of chemicals, formation of larger quantities of chemical sludge which is contaminated with heavy metals that are present in the added salts, increase the salinity of the effluent, need for the additional measures to obtain effluents with less than 1 mg phosphate when most stringent demands must be met in the near future. And for more quantitative aspects of chemical phosphate precipitation, there's another review by Megang Fopoli. So between 1965 and 1975, it became clear that conventional wastewater treatment systems would be induced to accumulate considerably more phosphate um, than required for the normal bacterial growth. And this phenomenon is called enhanced biological phosphorus removal. So this process is based on the storage of the phosphate in the microorganisms. Then they have taken an electron micrograph image of a thin section of Acinetobacter johnsoni containing one or two large black polyphosphate granules. The bar indicates, I think, 0.5 micron and the form of polyphosphate. So complexes of polyphosphate metals, proteins and nucleic acids dump together and form electron dense granules. The biology of this polyphosphate accumulating bacteria is discussed in several papers out there. Excellent reviews on the biological, biotechnological and ecological aspects of enhanced biological phosphorus removal and on the enzymes involved in the microbial polyp metabolism have appeared quite recently. However, the polyphosphate accumulating organisms that were isolated from sludges exhibiting enhanced biological phosphorus removal were largely neglected. Enhanced biological phosphorus removal, the history of this process is generally believed to have started with the first detailed lab analysis of the process in an activated sludge system by someone called Srinath et al. And these authors found that more phosphate was removed than could be accounted for by normal bacterial growth. Then Barnard et al. They discovered the treatment plants designed for nitrogen removal by nitrification denitrification systems were able to eliminate almost all phosphate as well. And the same year, it was found that an anaerobic period was essential for enhanced phosphorus removal. So, by introducing an anaerobic period in a conventional biological activated sludge process, enhanced phosphorus removal can be also achieved in a simple one vessel lab scale system. And in this so called fill and draw system, sludges with phosphorus contents up to 90% of the dry weight can be developed in a period of 6 to 16 weeks. When the system is started with the sludge that already possesses the ability to remove phosphorus to various degrees. And sludges from well buffer systems like the lab scale FND system do not contain chemically precipitated uh, phosphates. So the aerobic isolation of the pure cultures from any kind of sludge bacteria also reveals that 70 to 80% of the cultures accumulate polypi during aerobic growth or as a mineral salts medium with acetate as a sole carbon and energy source. So this is basically why this EVPR process is important. And various biochemical models have been proposed for the EVPR process, which tries to explain the metabolic transformations of PAOs. Basically, they agree on the major aspects. However, they differ on certain important aspects. Certain biochemical important aspects, and the main biochemical transformations that are widely accepted in the literature are all over the internet. You can just go and take a look regarding any kind of doubt that you have. So this statement was had the correct answer for this question was polyphosphate accumulating organisms and oxygen accumulating organisms. So these are the organisms that are involved in the biological phosphorus removal process. Moving on to the next question. Anybody? So I already showed the answer. It is phosphorus accumulating organisms. They assimilate volatile fatty acids and produce polyhydroxybutyrate storage products. Anybody has any kind of different opinion? So in the anaerobic zone, the PAO utilizes volatile fatty acids such as acetate or propionate to form polyhydroxyalkanoates which is a carbon storage compound, and the type of polyhydroxyalkanoid that is formed depend on the type of VFA that is used by the PAW, that is for polyphosphate phosphorus accumulating organisms. So these are basically fermentative bacteria. The VFAs are formed by fermentative bacteria in the anaerobic reactor, or are basically already present in the infant, and it can be beneficial to provide a pre-fermentation tank in the process configuration to enhance the levels of VFAs. Moving on to the next question. Which of the following is not considered part of an exposome? Anybody? Anybody? Which of the following is not considered to be a part of an exposome? If anybody knows what an exposome means, they can also say it.
Genetic factors.
Okay, this is not a part of exposome. Okay, anybody else? Try, guys. Anybody else, if you want to try? The answer question is not considered part of exposome. Okay, yeah, she's correct. So it is genetic factor. So I'll explain what an exposome is. So environmental factors taken to include environmental pollutants and lifestyle behaviors are main causes of various chronic non-communicable diseases that dominate worldwide mortality, including cardiovascular disease, cancer, respiratory disease. And the recent global burden of disease exercise has estimated that around 50% of all mortality worldwide was attributed to a few environmental factors, including indoor and outdoor air pollution, active and passive smoking, diet, and at the same time, the etiology of many common and highly complex chronic pathogens is still poorly understood. An example is asthma, in which complex interactions between environmental, social, and lifestyle factors, that is, ambient air quality, house dust, mold, smoking, and all the genetic and epigenetic factors are likely to underlie development and exacerbation. However, we have still not been able to measure environmental factors with the same degree of accuracy and comprehensiveness as the genome, and this due to the many uncertainties in assessment of the environmental exposures, which are traditionally measured through questionnaires and geographical mapping. Basically, under the lack of the studies that tackle multiple exposures, the environmental component is disease etiology has thus largely been studied using a one exposure, one health effect approach. So, what is an exposome? Anybody who would like to try talking about an exposome? It's invisible, right? My voice is audible? Yeah. Anybody would like to tell what an exposome is? Okay, so an exposome is basically the external. One second. Some issues. Yeah. So the exposome is basically first proposed by scientist Wild, to encompass the totality of the human environmental, meaning basically all non-genetic exposures from conception onwards, complementing the genome. So this concept was developed to draw attention to the need for better and more complete environmental exposure data in order to balance the investment, tools, and knowledge in genetics. So three overlapping domains within the exposome have been described as follows, like general external environment to include factors such as the urban environment, a climate factor, social capital, stress, a specific external environment with specific contaminants, diet, physical activity, tobacco infections, etc. Then an internal environment to include internal biological factors such as metabolic factors, gut microflora, inflammation, oxidative stress, etc. And there are large challenges in developing the concept of measuring every environmental exposure over a lifetime into a workable approach. A successful exposome would have to integrate many external and internal exposures from different sources continuously over the life course. But still, what is the external exposome? So, in the exposome concept, the challenge of accurate and reliable exposure assessment is multiplied because it requires obtaining exposure data for many exposure variables. And the example of asthma, this would mean an accurate and simultaneous measurement of many factors related to the indoor environment: air quality, air pollutants, pests, mold, dust, etc., and the outdoor environment like air pollution, green spaces, urban environment, as well as social and lifestyle factors. So, within the external exposome, a distinction can be made between exposure in the specific external environment, including tobacco smoke, diet, and chemical contaminants, which are traditionally largely assessed at the individual level by questionnaires, and exposures in the general external environment, such as outdoor air, pollutants, noise, and the urban environment, which are mainly assessed at the community level through geographical mapping methods. So the answer is genetic factors. Genetic factors do not belong the chromosome. So for individual assessed exposures, improvement in the exposure assessment can be achieved through better modeling of exposure. For example, by using predictive exposure models that combine questionnaire information with biomarkers and monitors for validation. For example, cotinine biomarkers may be used to validate the questionnaire responses on environmental tobacco smoke, and indoor air pollution monitors may be used to validate questions on cooking and heating sources in the home. So new smartphone linked diaries and imaging are also promising tools for more accurate and complete exposure assessment, for example of diet and use of consumer products. For community level exposures, major improvements can be achieved by improving information on where people are, how they move through their environment, and in case of air pollution, how much air they inhale. And knowledge about the inhalation rate, for example, may be integrated with personal air pollution measurements to estimate inhalation dose. For this information about physical activity is needed, and this can be collected through readily available accelerometers or sensors in smartphones. Further, people tend to move around in microenvironments. For example, near busy roads, where exposure such as air pollutants and noise can be considerably higher compared with the rest of the time, or in parks where exposure to green space is higher and exposure to air pollution and noise is lower. So, smartphone applications that integrate global positioning systems, location data with physical activity, information, and pollution measurements, and are now being developed to better characterize the external exposome. What is the internal exposome though? The high throughput molecular omics technique can actually analyze complete sets of biological molecules, including smaller molecules, metabolomics, larger molecules, proteome, gene expression profiles, transcriptomics, and epigenomics, and reactive electrophiles, adaptomics. So recent years have seen the rapid development of omics applications in animal and in vitro experimental studies. For example, there are now numerous reports of epigenetic, such as methylation and miRNA modifications arising from exposure to environmental toxicants. In human studies, applications have been extremely limited, but promising new results relate to their use to predict exposure-related health risks and individual susceptibilities. For example, methylation profiles in relation to air pollution and cigarette smoking, metabolomic profiles in relation to diet, and protein profiling in relation to benzene, arsenic, and lead. In the development of the exposome concept, the contribution of omics techniques is likely to lie mainly in their potential to measure profiles or signatures of the biological response to complex exposure mixtures or a cumulative exposure experience. In particular, the hope is to find an unique matrix that could play an equivalent role to the DNA sequence in genome-wide association studies and allow the characterization of the exposome without characterizing each exposure separately. And this is exactly the point where classical single exposure biomarkers have reached their limits. The measurement of numerous single analytes has limits in cost and available sample quantity. It is expected that the untargeted nature of omics data will capture biological responses to exposure in a more holistic way, provide clues about thus far unidentified risk factors, and give extra information on the molecular mechanisms underlying exposure related health effects. Further evaluation of omics tools for use in environmental health research requires careful attention to be paid to challenges relating to study design, validation, replication, temporal variance, and metadata analysis. For example, for these techniques to be useful in larger studies, it has to be shown that intra-individual variability in the molecular profiles measured in the systemic body fluids that can be collected in population studies like blood, urine, is less important than inter-individual variability. Profiles of RNA transcripts, proteins, and metabolites are highly variable over time. Teasing out variations due to exposure changes contains a major challenge. Then, what is a dynamic exposome? The exposome is dynamic as opposed to the static genome, and exposures vary on an hourly to yearly basis in the external and internal environments, basically. So, moreover, a given exposure or dose will not have the same effect during the various developmental periods that are critical to health and disease. Measurement of the exposome at any one given point at time would not be sufficient to characterize all health impacts of the environment, but there are currently no prospective study as such. Study populations that would allow lifelong repeated characteristics. So one way forward would be to start developing the exposome at certain key points in the exposure experience or disease development. For example, it is well recognized that the periods of organ development during prenatal life and infancy are especially vulnerable to the effects of environmental risk factors, which may manifest themselves throughout the lifetime in adult diseases, including respiratory and allergic diseases. Early life may thus be a key point for defining the exposome, and the efforts are underway to accomplish this using a prospective birth cohort studies. Another option is to apply the exposome concept to several cohorts and span the entire life course. So, how will you analyze the exposomes? How will you analyze the exposomes outcome association with health? So, collection of data on many chemical and physical exposures and on molecular omics profiles generates huge amounts of data which have to be stored, managed, analyzed, and interpreted. And statistical analysis of environmental risk factors have traditionally used regression models suitable for one exposure, one health effect type analysis. Approaches that may prove useful to evaluate multiple and combined exposures include the evaluation of risk estimates for many single exposures in a agnostic and hypothesis generating manner. The so-called environment-wide association study approach has been used to discover priority risk factors for type 2 diabetes mellitus and all that cause mortality. The evaluation of risk estimates for combined exposures through data driven dimension reduction methods, basically, and the evaluation of risk estimates for the groups of subjects sharing a similar exposome, such as in Bayesian profile regression analysis. So once risks related to the exposure have been estimated, systematic impact assessments of health effects are then needed to inform the development of policies, and health impact assessments aim at providing decision makers with like sound information on policy implications for health. And traditional policy development tends to be one dimensional, focusing on one or a limited number of risk factors and one or a limited number of health outcomes. So tools are basically now under development that try to capture and capture the complexity of multiple exposures and health outcomes and integrate risk and benefits in a more holistic model. So that is basically the entire concept of exposome. While genome is very static, exposomes are dynamic. As you can see, it will depend on the exposure to various internal and external factors as well. Whereas genome is a particular sequence that is very static in your body, it's not going to change. That is why genome is stable and exposome is very dynamic. And this schematic shows the it's basically a Venn diagram between a genome and exposome. So gene environment, there's a lot of interaction that occurs between the genome and the exposome. So any kind of disease that a person has can be hereditary. Definitely heredity is a fact, is a very huge has a very huge impact, because some diseases can be inherited. But exposome, like exposures to external agents, is also very prominent in certain diseases. I'm sorry if there's noise coming in from outside. So exposures to these agents is also very prominent, because at certain time point, like if we are talking about any kind of cancer, obviously it can be hereditary as well, but also exposure to regular routine smoking habits exposure to even passive smoking, if someone is smoking in front of you, exposure to certain agents that will carcinogens will also increase your risk towards cancer. So that's why exposome also plays a very prominent role in this. So is the noise disturbing? Am I audible over the noise? Can someone let me know? Can someone let me know if I'm audible, clearly audible over the noise that's coming from outside?
Yes.
Okay, okay. Thank you. Yeah, so that is why exposure. I'm really sorry for this. There's a lot of noise coming from outside. So that is why exposome. It's a very new concept. Like we have already heard about the omics concepts like proteome, genome, and this metabolome, transcriptome. Exposome is something that even I'm hearing for the first time. So it's a very different concept, but still very prominent in today's time. So that is why exposome is dynamic and genome is static basically. Moving on to the next question. This is another slide that depicts the interaction. This is ecosystems like food outlets, alcohol outlets. This is how the lifestyle is also very important in dictating any kind of disease that you might have. Physical activity, if you're having any kind of physical activity, sleep behavior, diet, drug use, smoking, then social interactions, if you're having any kind of social interactions, social networks, all of these are also very important. And believe it, even social interactions, your social way of living life will also have a very big impact on some certain diseases, mental, physical diseases. Physical chemical effects is all obviously important. Temperature, humidity, ambient light, anything, any kind of contamination, food contaminants, soil contaminants, pesticides, fragrance products, and all these. So this is basically the entire concept of exposome. Whatever you're exposed to, the entire thing is called exposome. So moving on to the next question, which of the following statements are true is or are true false? Statement A: biomarkers could be used to assess exposure to environmental toxins or other environmental factors. They can be measured in various biological samples such as blood, urine, or saliva. Statement B: by measuring the levels of certain biomarkers in the body, researchers can assess the extent of exposure to various environmental factors and evaluate the risk of developing a disease. Which one do you think is correct? Anybody? Anybody?
Both are true.
I think both are true. Anybody else? Yeah, correct. So both A and B are true. So what are biomarkers basically, and what do these biomarkers do? So biomarkers of effect are measurable, changes in an individual that indicate health impairment or disease. Although biomarkers have long been a very crucial part of the medical practice, blood pressure is a simple example. Basically, research have like how it's basically a kind of biomarker, right? Blood pressure checking, everything that's happening. Researchers have recently identified a variety of new biomarkers that signal the presence of conditions such as nervous system damage, autoimmune disorders, and cancer. And of particular interest is the potential of these new biomarkers to, measure adverse health effects that may arise from exposure to environmental pollutants. So. There was a very recent workshop that was held to basically explore. I think it was held in the US, and I don't remember the place. So the workshop basically was exploring how new biomarker approaches can be applied to understanding the consequences of environmental exposures and improve the environmental health decisions. So there, in the workshop, there were multidisciplinary groups, including experts in public health, environmental health, clinical medicine, and health disparities to discuss the state of the art of biomarkers and health. And I think it was the workshop was by the National Institute of Environmental Health Sciences. So there were presentations that were showing the different types of biomarkers and how everything is happening. So if you talk about the proper definition of a biomarker of effect is any measurable biochemical, physiologic or other alteration within any organism that depending on magnitude can be recognized as an established or potential health impairment or disease. And this terminology must be clearly explained to ensure that everyone is aligned on what is being discussed. So other terms are validation and qualification, which some people use quite interchangeably in the in the domain of biomarkers. So these are some of the prominent biomarkers, categories of biomarkers that are out there, and what are the roles of these biomarkers. So what are diagnostic biomarkers used for? Diagnostic biomarkers are used to confirm the presence of any disease or any subtype of any disease or medical condition. That's how, like, people have certain, they can actually assess the levels of certain substances in their body to figure out if a particular disease, if they are infected, if they are suffering from a particular disease or not. So obviously a particular disease will hamper the normal signaling pathways, it will hamper various things in your body, and that will lead to various changes of substances in your body. So the presence of any kind of particular substance that is only prevalent in the diseased patients will help you confirm that that patient has a particular disease. That is the role of diagnostic biomarkers. Really sorry for the noise. Anyway, that is the role of the diagnostic biomarker. If at any point you're not able to hear me over the noise outside, please let me know. Anyway, I'll be uploading all these videos on YouTube and the links will be shared, so you can actually check the videos out later also. But still, I'm really sorry for the noise that's coming from outside. It's not under my control. Anyway, so these are the roles of the diagnostic biomarkers. Then there's a category called monitoring biomarkers. So in monitoring biomarkers, what happens is that it basically assesses the present status or extent of a disease or medical condition to evaluate the response to the intervention. So basically, they're monitoring the extent of the disease. They're monitoring the status of a disease. So now you know that the patient has a disease. You have confirmed that the patient is having the disease because of the presence of a diagnostic biomarker. But now you have to monitor how that biomarker is. What is the status of that biomarker? What is happening basically, to the disease progression is happening or not. So that is what is done using the present status or extent of any particular disease or medical condition. Then we have the pharmacodynamic or response biomarkers. So it basically helps to evaluate the response to a medical condition or clinical intervention. So that is what response biomarkers is all about. I have not heard about this particular category a lot. I've mostly heard about diagnostic biomarkers, which is very prominent in any kind of medical or public health field. Then there are predictive biomarkers. So identify the probability of developing a clinical event after an exposure to a medical product or environmental agent. So nowadays, people, after the huge success of the Human Genome Project, there were people who actually got their entire genome sequenced, and they wanted to know if they had a particular, high chances of getting a particular disease. So if someone from a family suffering from Alzheimer's, the offsprings, the next generation of the family can actually get their entire genome sequence and find out if they do have a high chance of getting that particular disease. So that is, I know a lot of Hollywood actors who have actually done this. So they have sequenced their genome, they have tried out different techniques to figure out if they are more prone to a particular disease than others. So I think predictive biomarkers is kind of like that only. It can help you identify the probability that you have of developing a clinical event, positive or negative, after the exposure to a medical product. So I don't know the exact procedure how it's carried out. I'm pretty sure it's very optimized and different for different diseases, different for different systems. But what happens is that you get exposed to a particular agent, and then you try to figure out people try to figure out the probability of developing a particular disease based on the outcome that you are showing. Then we have prognostic biomarkers. So it helps in identifying the likelihood of a clinical event, disease recurrence or progression in patients diagnosed with a disease or having a medical condition. So some patients are already diagnosed with a particular condition, a particular disease. So this type of biomarker will help you identify the likelihood of the disease progression in a particular manner. So these are different category of prognostic biomarkers, still very popular and very prominent in the medical and the health field. Then you have the susceptibility or the risk biomarkers, quite quite similar to the predictive biomarkers. It measures the risk of an individual to develop a disease or medical condition in patients without the disease or medical. So someone who has still not developed the disease, or who is still not suffering from any kind of medical condition, can actually get tested with the help of some risk biomarkers, susceptibility biomarkers, and they can actually figure out the risk that they possess, that an individual has in developing the particular disease. Then we have the safety biomarkers. So what are safety biomarkers? In safety biomarkers, it basically helps to predict toxic adverse events induced by drug, medical intervention, or any kind of environmental agent exposure. That are safety biomarkers. Okay, so moving on to the next question. Which of the following is a primary building block of proteins? It's very easy and basic. Anybody?
Amino acid.
Yeah, correct. Anybody else? Yes. Yeah, it's pretty basic. So it's amino acids, and we know that proteins, the amino acids, the building blocks of proteins. So the other options were antibodies, pigments, and terpenoids. Definitely not. Amino acids are the primary. Amino acids basically form the primary level of primary structure of proteins. So as we know that proteins are end products of the decoding process that starts with the information in cellular DNA. And as workhorses of the cells, proteins compose structural and motor elements in the cell, and they serve as the catalyst for virtually every biochemical reaction and that occurs in living things. So this incredible array of functions that derives from a startlingly simple code that specifies a hugely diverse set of structures. So in fact, each gene in cellular DNA contains the code for unique protein structure. So not only are these proteins assembled with different amino acid sequences, but they are also held together by different bonds and are folded into a variety of three-dimensional structures. So the folded shape or conformation depends directly on the linear amino acid sequence of the protein. So what are these proteins made of? The building blocks of proteins are amino acids, which are small organic molecules that consist of an alpha central carbon atom linked to an amino group, a carboxyl group, a hydrogen group, and a variable component called a side chain. So within a protein, multiple amino acids are linked together by peptide bonds, thereby forming a long chain. And peptide bonds are formed by a biochemical reaction that extracts a water molecule as it joins the amino group of one amino acid to the carboxyl group of a neighboring amino acid. So the linear sequence of amino acids within a protein is considered the primary structure of the protein. Proteins are built from a set of only 20 amino acids, each of which has a unique side chain. And the side chains of the amino acids have different chemistries. And the largest group of amino acids have non-polar side chains. Several other amino acids have side chains with positive or negative charges, while others have polar but uncharged side chains. The chemistry of amino acid side chain is critical to protein structures because these side chains can actually bond with one another to hold the length of protein in a certain shape or conformation. Now, charged amino acid chains can form ionic bonds, and polar amino acids are capable of forming, hydrogen bonds. Hydrophobic side chains, they interact with each other via weak van der Waals interactions, and the vast majority of the bonds formed by these side chains are non-covalent. In fact, cysteines, I think, are the only amino acids capable of forming covalent bonds, which they do with their particular side chains. Because of side chain interactions, the sequence and location of amino acids in a particular protein guides where it bends and folds occur in that particular protein. So the defining feature of an amino acid is its side chain. When connected together by a series of peptide bonds, amino acids form a polypeptide, another word for protein. Like polypeptides also, they fold accordingly and form protein. There are different subunits that might come and actually fold together to form protein. The polypeptide will then fold into a specific conformation depending on the interactions between its amino acid side chains. So that is what the primary structure is all about. most amino acid sequence that is, it drives the folding and the intramolecular bonding of the linear amino acid chain, which ultimately determines the protein's unique three-dimensional structure and shape. And hydrogen bonding between the amino groups and the carboxyl groups in neighboring regions of the protein chains sometimes causes certain patterns of folding to occur. So these are known as the alpha helices or the beta sheets. These stable folding patterns make up the secondary structures of the protein. So most proteins contain multiple helices and sheets in addition to other less common patterns. And the ensemble of formations and folds in a single linear chain of amino acids, sometimes called a polypeptide, it constitutes the tertiary structure of the protein. Finally, the quaternary structure of a protein refers to those macromolecules with multiple polypeptide chains or subunits. And the final shape adopted by a newly synthesized protein is typically the most energetically favorable one. So, as proteins fold, they test a variety of conformations before reaching their final form, which is unique and compact. And folded proteins are stabilized by thousands of non-covalent bonds between amino acids, and in addition chemical forces between a protein and its intermediate immediate environment contribute to protein shape and stability. For example, the proteins that are dissolved in the cell cytoplasm have hydrophilic chemical groups on their surfaces, whereas the hydrophobic elements tend to be tucked inside. In contrast, the proteins that are inserted into the cell membranes display some hydrophobic chemical groups on their surface, specifically in those regions where the protein surface is exposed to membrane lipids. So it is important to note, however, that fully folded proteins are not frozen into shape. Rather, the atoms within these proteins remain capable of making small movements. Yes.
Backside noise is coming.
Yeah, 1 second. I give me a 10 minutes. I'll try to figure something out. 1 second. I'm really sorry for the sound disturbance. Is it better now? Can you hear me? Is it better?
Yes.
So actually, we live in hostels and there's probably an event going on, so that's why there's a lot of noise. So sorry for the disturbance. I'll share my slide again. Yeah. So yeah, so what I was saying is about protein structure. So the final shape that a protein, newly synthesized protein adopts is typically the most energetically favorable one. As I said, the primary structure will be the amino acid sequence, which will drive the folding and the intermolecular bonding of the linear amino acid chain, which ultimately determines the protein's unique three-dimensional shape. The hydrogen bonding between the amino groups and the carboxyl groups in the neighboring regions of the protein chain sometimes causes certain patterns of folding to occur. This will be called these are called the alpha helices and the beta sheets. And these stable folding patterns make up the secondary structure of a protein. And most proteins contain multiple helices and sheets in addition to their other less common patterns. And the ensemble of formations and folds in a single linear chain of amino acids, sometimes called a polypeptide, constitutes the tertiary structure of a protein. Finally, the quaternary structure of a protein refers to those macromolecules with multiple polypeptide chains or subunits. Now, the final shape adopted by a newly synthesized protein is typically the most energetically favorable one. As proteins fold, they test a variety of conformations before reaching their final form, which is unique and compact. Folded proteins are stabilized by thousands of non-covalent bonds between the amino acids. And in addition, there are chemical forces between a protein and its immediate environment, which contributes to protein shape and stability. For example, the proteins that are dissolved in the cell cytoplasm have hydrophilic, which is water loving chemical groups on their surfaces, whereas their hydrophobic or water averse elements tend to be tucked inside because they are not coming in contact with any aqueous environment. In contrast, the proteins that are inserted into the cell membranes, they display some hydrophobic chemical groups on their surface, specifically in those regions where the protein surface is exposed to membrane lipids. Now, it's important to note, however, that fully folded proteins are not frozen into shape. Rather, the atoms within these proteins remain capable of making small movements. So, even though proteins are considered macromolecules, they are still too small to visualize, even with a microscope. So, scientists must use some kind of indirect method to figure out what they look like and how they are folded. So, the most common method used to study protein structure is called X-ray crystallography. And with this method, solid crystals of purified proteins are placed in an X-ray beam, and the pattern of the deflected X-rays is used to predict the positions of the thousands of atoms within the protein crystal. So, how do these proteins arrive at their final shapes? So, in theory, once their constituent amino acids are strung together, proteins attain their final shapes without any energy input. In reality, however, the cytoplasm is a crowded place, as we all know, filled with many other macromolecules capable of interacting with a partially folded protein. So, inappropriate associations with nearby proteins can interfere with proper folding and cause large aggregates of proteins to form in cells. So, cells therefore rely on so called chaperone proteins to prevent these inappropriate associations with unintended folding partners. So, what are chaperone proteins? Chaperone proteins are nothing but they are basically helper proteins. They surround the protein during the folding process, sequestering the protein until the folding is complete. For example, in bacteria, multiple molecules of the chaperone GROEL form a hollow chamber around proteins that are in the process of folding basically. So molecules of a second chaperone called the GROES or GROS then form a lid over the chamber, and this is what happens in bacteria. For eukaryotes, they use different families of chaperone proteins, although they function in similar ways. So, chaperone proteins are also very abundant in cells. These chaperones use energy from ATP to bind and release polypeptides as they go through the folding process, and chaperones also assist in the refolding of proteins in cells. So, folded proteins are actually very fragile structures which can very easily denature or unfold. So, although many thousands of bonds hold proteins together, most of the bonds are non-covalent and fairly weak. So, even under normal circumstances, a portion of all cellular proteins are unfolded. And increasing body temperature by only a few degrees can significantly increase the rate of unfolding. So when this happens, repairing existing proteins using chaperones is much more efficient than synthesizing new proteins. Also, interestingly, cell synthesis additional chaperone proteins, which are also the HSP family of proteins or the heat shock proteins. Now, what is a protein family? I just mentioned HSP family. So, all proteins bind to other molecules in order to complete their tasks, and the precise function of a protein basically depends on the way its exposed surface interacts with those molecules. So, proteins with related shapes tend to interact with certain molecules in similar ways, and those proteins are therefore considered to be belonging to a same protein family. So the proteins within a particular family tend to perform similar functions within the cell. Proteins from the same family also often have long stretches of similar amino acid sequences within their primary structure. So these stretches have been conserved through evolution and are vital to the catalytic function of the protein. For example, cell receptor proteins contain different amino acid sequences at their binding sites, which receive chemical signals from outside the cell, but they are most similar in amino acid sequences that interact with common intracellular signaling proteins. So, protein families may have many members, and they likely evolved from ancient gene duplication events. And these duplications finally led to modifications of the protein functions and expanded the functional repertoire of organisms over time. So, in conclusion, proteins are built of chains of amino acids which then fold into unique three-dimensional shapes, and bonding within protein molecules helps stabilize their structure, and the final folded forms of proteins are well adapted for their functions. So, even though it might seem very simplified, it is not. Protein structures are very complex, and the primary structure might be just a simple length of amino acid chain, but in reality proteins and any working with proteins is very difficult. When you are working in the lab, getting crystals for proteins, doing extracrystallography, figuring out the structure of proteins, it's very difficult. That's why the papers which actually discover new structures of proteins, they actually published in very high impact factor journals, because the work is not easy. The primary sequence might be just a simple sequence of amino acids, but as and when the protein folds, secondary, tertiary, and finally quaternary structure, deciphering the structure of a protein is very difficult and very challenging, even to till this date, when science has advanced so much and we have so many instruments at our disposal. So yes, you are correct. The primary building block of proteins is amino acids. Uh, she's correct. So moving on to the next question. Yeah. So statement A is increase in the levels of antibiotic resistance is triggered by presence of environmental contaminants such as pesticides and pharmaceuticals. Statement B is antibiotic resistance is fast increasing in pathogens while the discovery of new drugs is decreasing. Which statement is true? Anybody? Anybody would like to answer? Anybody? I'm audible, right? Sorry, there's internet issues also. Yeah. Yes, I am. Is anybody who would like to answer? Okay, so A and B are both true statements. You can see the answer. So, what is antibiotic resistance? So, antibiotic resistance is a global phenomena that has been, hampering, like, uh, like you must have heard about multi-drug resistant bacteria, XDR bacteria, MDR bacteria. So, all these terms come from antibiotic resistance. So it's a global health challenge involving the transfer of bacteria and genes between humans, animals, and the environment. And although multiple barriers restrict the flow of both bacteria and genes, pathogens recurrently acquire new resistance factors from other species, thereby reducing our ability to prevent and treat bacterial infections. Evolutionary events that lead to the emergence of new resistance factors in pathogens are rare and challenging to predict, but may be associated with vast ramifications. So transmission events of already widespread resistant strains are, on the other hand, common, quantifiable, and more predictable, but the consequences of each event are limited. So quantifying the pathways and identifying the drivers of and bottlenecks for environmental evolution and transmission of antibiotic resistance are key components to understanding and managing the resistance crisis as a whole. So there are a lot of review papers online where the authors present the current understanding of the roles of the environment, including antibiotic pollution in resistance, evolution in transmission, and as a mere reflection of the regional antibiotic resistance situation in the clinic. So they provide with evidences, they describe risk scenarios, they discuss methods. So if you're fascinated by this like I am, I think you should go online and read some of the excellent reviews that are present, that are published. So antibiotic resistance can arise both from mutations in the pre-existing genome of a bacteria and from the uptake of any kind of foreign bacteria. So mutations readily occur and become fixed in the patient or animal treated with the antibiotic. Such a strong selection pressure on pathogens is rare elsewhere. So the process is also independent of the genetic reservoir in other species. So hence external environments are generally less likely to provide a major contribution to the mutation-based evolution of resistance for most pathogens. So with regard to uptake of novel resistant factors, water, soil and other environments with highly variable ecological niches provide an unmatched gene pool with a diversity that greatly exceeds that of the human and the domestic animal microbiota. Indeed, the most striking feature of the environmental microbiome is its immense diversity, providing numerous genes that potentially could be acquired and used by pathogens to counteract the effect of antibiotics. All approved antibiotic classes so far, whether they be natural, semi-synthetic or synthetic compounds, have been met by resistance in at least some of the pathogens they target. So this suggests that external environments already harbor resistance factors for all antibiotics that will ever be developed, unless we start thinking radically different about how antibiotics are designed. So over the years, most antibiotic resistance genes have probably evolved gradually from genes with other functions. So the more recent evolutionary events responsible for their widespread occurrence in pathogens are mainly a result of transfer events from ancestral species in which the overall functionality of the genes were shaped. Starting from a chromosomal immobile antibiotic resistance genes, there is typically a stepwise evolution that leads to acquired resistance in a pathogen. So the first step is often the ability of an antibiotic resistant gene to move within the genome, attained, for example, by association with insertion sequences or the formation of gene cassettes and incorporation into the integrons. The second step involves the relocation of that gene to an element that can move autonomously between cells, such as plasmids or an integrative conjugative element. So some environments are probably more likely than others to provide the various genetic elements typically involved in mobilization and transfer of antibiotic resistant genes, either through the presence of fecal bacteria known to often carry such elements, or possibly because the conditions favor frequent gene exchanges. The third step is the horizontal transfer of a mobilized resistance gene, either directly to a pathogen or via one or several intermediary bacterial hosts. So the fourth step, which may also occur at any time in the process, is the physical transfer of the bacterium carrying the antibiotic resistance gene to the human or domestic animal microbiota. And this ability is described by the term ecological connectivity. High metabolic activity and extensive cell-to-cell contact, such as in biofilms, are probably increasing the rate of most steps. So all of these steps, including mobilization, for example, insertion sequences or integrons, increase in donor cell abundance and thus transfer opportunities and the rate of horizontal gene transfer. And it may be promoted by antibiotics. Importantly, though, most, if not all, the steps also occur in the absence of antibiotics, but at different rates. Hence, it's very crucial to understand where the bottlenecks are in the evolution towards resistance in pathogens. A critical bottleneck is likely to be the selection of the rare genotypes with acquired resistance that result from mobilization or horizontal gene transfer, genotypes that otherwise would disappear. At all stages, compensatory mutation somewhere in the genome of the bacterium carrying the antibiotic resistance gene may occur, lowering potential fitness costs either through reducing niche overlap or by increasing competitive ability. And the emergence of new antibiotic resistant genes in the clinic occurs only when the events align in time and space. So, antibiotic resistance it's a very critical factor in today's time, and it has actually led to the development of many antibiotic resistance bacteria that are it's very difficult to treat. So people sometimes start antibiotic dose and they don't without consulting doctors, which is very wrong. You should always consult a professional before starting any kind of antibiotic dosage. And often when even when it's recommended by a doctor, people start the dose but they don't finish up with the dose. So what happens is that when you start the antibiotic dose, you start killing, if it's showing any kind of bactericidal activity, you start killing the bacteria in your body. But if you don't finish the dose that has been recommended to you, then the bacteria in your body, they no longer die from it, they become resistant to it. And then the susceptible bacteria, they all die out, only the resistant bacteria stay. And that's how antibiotic resistant bacteria come into existence. So that's why only take antibiotics when doctors or professionals recommend it to you. And if you start the dose of any dosage of any antibiotic, please have the patients to complete the entire dosage. Otherwise this antibiotic resistance is a huge huge challenge in today's world. There are a lot of bacteria, mycobacterium tuberculosis, all of these have become multi-drug resistant. So at some point of time it will be impossible to treat this bacteria. People are trying out new techniques, science is advancing, and still this is a very huge topic. And it's still a very hot topic, but I think in the coming 2-3 years it will be the most studied and most researched topic ever, because this is a huge concern. It's a very challenging thing to actually make the antibiotic resistant bacteria susceptible to antibiotics again. So yeah, so that is why it's truly very fast increasing in any kind of pathogen. And the discovery of new drugs is not decreasing, I'll say it's stagnant. It's very difficult to discover a new drug. It's not really a 2-minute task. So that's why both these statements are true. So moving on to the next question. Okay, so this was a schematic that whatever I explained, this is a schematic that basically shows it in a diagram form basically. One second. Yeah. So chromosomal immobile antibiotic resistance gene is coming from the environment, then mobilization within the genome in integrons, relocation to an element that can move autonomously between cells like plasmids or conjugative elements, then horizontal transfer of such a mobile element either directly to a pathogen or via one of the several intermediary hosts. So horizontal gene transfer can happen by the process of conjugation. So plasmid, F plasmid can get transferred to another bacteria either in the normal environment or when both the bacteria are infecting a particular host, so via the intermediary host. And that is how the entire antibiotic resistance gene is coming into existence, and the thing is proliferating more and more. Yes, so moving on to the next question. Which of the following mechanisms of horizontal gene transfer is carried through a bacteriophage that infects a bacterial cell? So I've discussed similar questions before. Anybody would like to answer? Anybody? Audible, right? You guys can hear me, right?
Conjugation.
So bacteriophage, you think it's conjugation? Anybody else? It's not conjugation, it's transduction. So there are three horizontal gene transfer techniques: transformation, conjugation, and transduction. Transformation in when is the foreign DNA is taken up by the bacteria. Transformation is what we do in labs. In labs, when we want to genetically engineer a particular plasmid with our particular gene of interest, we insert that gene of interest, for example, a protein that we want to produce in huge amounts. So we have the gene for that protein, we incorporate it into the plasmid, we transform. So now the gene is within the plasmid. So now then we have to transform the plasmid into the bacterial cell. Transform the plasmid. Sorry. Is anybody saying something? Yeah, so you have to transform the gene into the bacterial cell. So that is how basically there's a protocol for transformation. So you are transforming the plasmid containing your gene of interest into the bacterial cell, and now you're letting the bacteria grow. Uh, and then the bacteria will multiply, and ultimately when you are producing the protein, you'll get the protein in a huge amount. That is transformation. Conjugation is what is I discussed as previously only. Conjugation is when there's a F plasmid or a kind of plasmid, gets transferred from one bacteria to another. It is a very prominent type of horizontal gene transfer, and basically, well, wait, I have a picture. I think not in this one. Wait, I'll show you a picture of conjugation. One second. Give me a minute. Give me a minute. I'll show the three different types of horizontal gene transfer I had showed in a previous class, I believe. I don't remember which class. Give me a minute. Okay, it's not this class. I believe I had shown in some previous class. I'll just show the diagram. Three types of. Which class was it? Give me a minute. Yeah, I found it. Yeah. So I'll share my screen. It's an old slide. So it's an old slide. Yeah. So this is what transformation is. A bacteria, this is the bacteria, this is the chromosomal DNA, plasmid is getting transformed. So bacteria takes in the DNA from its environment, and often the DNA that's being shed by other bacteria. So that's how it occurs in the environment. In lab, the DNA may be introduced by scientists, what I was talking about. We add the gene of interest into the plasmid, and then we transform the plasmid into the bacteria. And if the DNA is in the form of a circular DNA, it's called a plasmid. So this is the transformation. I'll come to this is conjugation. Conjugation is basically DNA being transferred from one bacterium to another. So this is the donor cell, this is the recipient cell. The screen is visible, right? I'm not sure, I didn't ask anyone. Screen is visible, right? You can see the screen.
Yes.
Yeah. So this is how conjugation occurs. So this is the donor cell and this is the recipient cell. So the donor cell pulls itself close to the recipient cell using a structure called the pilus or the pillars, whatever you want to call. Then DNA is transferred between the cells, and in most cases this DNA is in the form of a plasmid. So donor cells typically act as donors because they have a chunk of DNA that have the fertility factor or the F factor. That's why it's sometimes called the F plasmid. So this chunk of DNA codes for the proteins that make up the sex pilus. So it also contains a special site where DNA transfer during the conjugation begins. And if the F factor is transferred during conjugation, the receiving cell turns into a F++ donor that can make its own pilus and transfer DNA to the other cells. So this is the process of conjugation. Now what was our question? Our question was which of the following mechanisms of horizontal gene transfer is carried through a bacteriophage that infects a bacterial cell? So the answer is transduction. So what happens in transduction? This is what transduction is. So bacteriophages are nothing but viruses that infect bacterial cells, not anything else like bacterial cells. So in transduction, the viruses or the bacteriophages infect the bacteria, they move short pieces of the chromosomal DNA from the bacteriophage into the bacterium. So the viruses, the bacteriophage, then like these are basically viruses for the bacterial cell. That's what they have said. And there are two cycles. I have talked about this in another lecture, lytic and the lysogenic cycle. So the bacteriophage then has to decide which cycle it will go into. Either they can go via the lytic cycle. So what happens in the lytic cycle? The bacteria will basically lyse. So bacteria will lyse the host cell. The not bacteria, sorry, the bacteriophage. The bacteriophage bacterial virus or the bacteriophage will lyse the bacterial cell if it's following the lytic cycle. If it's following the lysogenic cycle, then what will it do? It will incorporate its DNA inside the bacteria, and that particular bacteriophage DNA will go and sit within the bacterial host DNA. It will be called prophage. It will stay in a dormant state. It will get incorporated into the bacterial uh, genetic material, and then that is called the lysogenic phase. And there is a very great explanation online where you will see how the bacteriophage decides whether to go to lytic or the lysogenic phase. So it's basically a very complex operon kind of thing. If you're interested, you can actually go online and search and read about it. What nutrients, what environment, how, like what are the external factors that help the bacteriophage decide whether it will go by the lytic phase or the lysogenic phase. It's very complicated. It might sound easy, but it's not. So but the basic thing is that there are two phases, lytic and lysogenic phases. So that this process is called transduction, not conjugation. So this is the basic procedure of transduction. You can see the screen. So bacterial transduction, the bacteriophage is coming, it is incorporating its DNA inside, then the DNA is going inside, it is replicating, it is basically hijacking the host machinery to form its own assembling particles. The assembling particles are formed, the bacteriophage is assembled inside the bacteria, then lysis is happening. Or else, this is the other thing, or else the bacteriophage is incorporating its DNA inside. Sorry. This is a continuation. This is just showing a very general schematic. They are not talking about lytic or lysogenic cycle. So this is a very genetic schematic. So now this thread, the blue thread is the bacterial chromosome, the host chromosome, and the pink thread is the bacteriophage chromosome. So the bacteria incorporates the DNA inside, it hijacks the bacteriophage, incorporates the DNA inside the bacterial cell, it hijacks the bacterial host machinery, and then various bacteriophage particles are assembled with the genome inside. Some bacteriophages have their own genome, some bacteriophages have the combined genome, and that is then this bacteriophage, that is the cell getting lysed, lytic cycle. Cell is getting lysed, the bacteriophages are released into the environment. These bacteriophages go and infect a new cell, and then it incorporates its DNA inside the new cells. This is how the bacterial transduction process is happening. So you can actually find very cool videos on YouTube also. If you are interested, you can go and search bacterial transduction mechanism shown in detail. So the answer for this question is transduction. Moving on to the next question. Which parameter is not suitable for selection of antibiotic resistance? Anybody? Okay, so the answer is antibiotic resistant microbes are non-proliferate. This statement is not suitable. This parameter, this statement, if true, is not true, but if true, is not suitable for selection of the antibiotic resistance. So there will be two kinds of bacteria at some point: antibiotic resistant and antibiotic susceptible. So if the antibiotic resistant microbes are not proliferating only, then they will not survive in the environment. So that will then the environment the system is not selecting for antibiotic resistance. If the antibiotic resistant microbes are not proliferating, that means the antibiotic resistant microbes will at some point die out. the antibiotic susceptible microbes are surviving. So at some point, the antibiotic resistance will die out. There will be no selection for antibiotic resistance. So that's why this statement is not suitable. So to read that, basically we know that antibiotics are a vital component of global health. By killing or inhibiting the growth of bacteria, antibiotics can treat infections like pneumonia, staphylococcus, tuberculosis, etc. So by preventing infections, they enable major medical procedures such as surgeries and chemotherapy. However, bacteria are still becoming increasingly resistant to current antibiotics, and so antibiotics are drugs that kill or inhibit the growth of bacteria, bactericidal and bacteriostatic, and they, they have been killing bacteria since when they were discovered. So penicillin was the first antibiotic that was discovered in 1928, but it was not until the World War II, when wounded soldiers were more likely to die from infections than the injuries themselves, that governments realized that penicillin is a life-saving drug. And then the government began developing and mass producing penicillin through unprecedented public, private, and international collaborations or whatever. So antibiotics are now used to treat a myriad of common infections like strep throat, meningitis, tuberculosis, tetanus, urinary tract infections, food poisoning. So they also enable medical procedures that otherwise create a high risk of infections, such as invasive surgery, organ transplantation, and chemotherapy. But antibiotics are not one size fits all. Certain types of antibiotics are only effective against certain kinds of pathogens, certain kinds of bacteria, and all antibiotics are ineffective against viruses. So antibiotics, they kill or inhibit bacterial growth via various mechanisms of action. They might attack the protective bacterial cell wall, interfere with the bacterial production, or interrupt production of molecules necessary for the bacteria's survival. However, bacteria reproduce and evolve rapidly, changing over time to resist an antibiotic's destructive mechanism of action. In fact, the more we use antibiotics, the faster bacteria evolve to resist those antibiotics, which is why it is not recommended to, get antibiotics over the counter and like swallow it whenever you feel like it. That's why it's very important to get a doctor's prescription before you can actually buy and swallow antibiotics. So in the more we use antibiotics, the faster bacteria evolve to resist those antibiotics. And as bacteria reproduce, when bacteria is reproducing, random DNA mutations will occur. Most random mutations have no effect on the bacteria, but sometimes a mutation will give the bacteria a special ability to resist an antibiotic. For instance, the mutation may change the cellular target of the antibiotic or allow the bacteria to pump the drug out of the cell. So when an antibiotic is used on bacteria, most of the population of the bacteria will die. But if any of the bacteria have one of those resistance conferring mutations, they will survive and continue to reproduce until the entire population is wiped out. The entire population is resistant. The use of antibiotics therefore creates an environment where bacteria with antibiotic resistant mutations are more likely to survive and reproduce, while susceptible bacteria are gradually killed off. So this figure shows use of antibiotic gradually increases the prevalence of resistant bacteria, and if any cell has developed characteristics allowing it to resist attack by an antibiotic, it is more likely to survive and multiply. This means that over time, the bacteria that cause infection in humans are more and more likely to be resistant to common antibiotics. It is important to note that bacteria develop antibiotic resistance, not people. But when people use lots of antibiotics, they change bacterial populations such that more and more bacteria are resistant to those antibiotic drugs. This illustrates basically the double-edged sword nature of antibiotic use. Antibiotics are immensely valuable for combating countless infections and enabling medical procedures, but the more we use them, the less valuable they become. So that is why even the antibiotics have made it possible to treat a variety of bacterial infections, even meningitis, bacteremia. Prior to their introduction, these were untreatable and consequently fatal. Unfortunately, in recent decades, overuse and misuse of antibiotics as well as social and economic factors basically have accelerated the spread of antibiotic resistant bacteria, making drug treatment ineffective. Currently, at least 700,000 people worldwide die every year due to antimicrobial resistance. So, without new treatments or new ways to combat this antimicrobial resistance, this will not work out. So although antimicrobial resistance is a very natural process, the public health emergency due to the uncontrolled spread of this phenomenon depends primarily on the overuse of antibiotics. What I was saying, we just think we have got a bacterial infection, we just buy it from the over the counter and swallow it. So you don't have the necessary knowledge to figure out if this is actually required or not. So that's why overuse and misuse of antibiotics have led to this antibiotic resistant bacteria. However, other factors are also primarily responsible for the increase in its prevalence, and there are various factors that I would not like to, would not, it's not possible for me to get into. Mostly I'll talk about the uh, the medical health aspect. So at one point when microorganisms stop responding to antibiotics, that is when the previously susceptible and previously active in treating infections, antibiotics become ineffective, and the bacterial infections become bacteria become resistant. So the statement was antibiotic resistant microbes are non-proliferating. Doesn't make any sense. If the antibiotic resistant bacteria become non-proliferating, then obviously they are not reproducing. Then the resistance antibiotic resistance selection will not happen. So for this particular question, this is the correct answer. Moving on, this is how antibiotic resistance spread. It can spread via mutation, via vertical transmission or horizontal transmission. So mutations occur. I already talked about the mutation aspect. Sorry, I've already talked about the mutation aspect. Vertical transmission is basically bacteria reproducing, and the offsprings are also getting the same antibiotic resistant genes. Horizontal transmission is what I discussed just now: transformation, conjugation, and transduction. Mostly it's conjugation, most likely, but transformation and transduction are also very prominent examples of how antibiotic resistance spreads. So the plasmid that is being transferred has the antibiotic resistance gene, and the new bacteria, the recipient bacteria, is also now getting resistant to the drug. So this is these are sorry, I'm really sorry. So this is these show some of the main mechanisms of antimicrobial resistance in escape pathogens. So these are the main four mechanisms. A drug, this is antibiotic drug, beta lactamase. So inactivation of the drug might occur, or drug target alteration. So a drug has been designed corresponding to a target, right? So the drug will go and bind to a target and then show its action. So the drug is designed according to that, but the bacteria now develops strategies to change the drug target structure by altering it. So now the drug is no more going and attaching to the target, and the effect of the drug is not safe. It's becoming ineffective. That's another mechanism of drug antimicrobial resistance. Then we have decreased drug uptake by alteration of outer membrane permeability. So previously the drug used to get inside very easily because of the more permeable bacterial membrane, but now the outer membrane permeability has altered, and that has led to decreased drug uptake. Then there will be drug efflux pumps activation. So once the drug goes inside, there are efflux systems that basically pump out the drug from inside the bacteria, basically saving the bacteria. These are the four most prominent ways of mechanisms of antimicrobial resistance in escape pathogens. So this is very interesting. As I said, in the next 2-3 years, I personally feel this will be the most hot topic to be ever researched, because it's a very challenging thing. Imagine if the antibiotics stop working, all these bacterial infections around us, and we have no way to treat them. So it's a very challenging thing that the scientists are struggling with each and every day. So these are the four most main mechanisms of antimicrobial resistance. So moving on to the next question. Ability to resist a toxin because of genes encoded in the same plasmid as antibiotic resistant gene is called cross resistance, co-resistance, co-regulation, co-expression. Anybody?
Cross resistance.
Okay. Anybody else? No, it's called co-resistance. So there are terms called co-selection, co-resistance, cross-resistance, and all of these. So this schematic basically shows what all this means. So co-selection of antibiotic and metal resistance in bacteria is important primarily because it can maintain and promote antibiotic resistance in bacterial populations in the absence of antibiotics. Co-selection is mainly caused by cross or co-resistance mechanisms. Now what happens? Cross resistance occurs when a single mechanism, that is an efflux pump for example, provides resistance to different compounds simultaneously. In contrast, co-resistance occurs when two or more different resistant genes are physically co-located on the same genetic element, such as a plasmid or a transposon, or are present in the same bacterial strain, where each resistance gene provides resistance to different compounds. So, in addition to co and cross resistance, co regulatory mechanisms can also promote the co selection process. So, this occurs when multiple resistance genes um, that confer resistance to different toxic compounds are controlled by a single regulatory gene. For example, the regulatory protein CZCR regulates the expression of the CZCCBA efflux pump, which confers resistance to zinc, cadmium, and cobalt. So this CZCR also co-regulates resistance to carbapenems. I'm not sure about pronunciation. I think carbapenems, which is a class of last resort antibiotic, by repressing the expression of the OPRD porin, the route of entry for these antibiotics to the bacterial cell. So this is the basic difference. This is the basic difference between the co-resistance and the cross-resistance mechanisms. You can see from the schematic also. These are the mechanisms of cross-resistance, co-regulation, co-expression of metal and antibiotic resistance. Cross-resistance happens when one resistance system confers resistance to both an antibiotic and metal. You can see this. You can see this. So this is cross resistance. One resistant mechanism is followed, for example drug efflux, and this system is both giving resistance to both the metal and the antibiotic, for example. Antibiotic, for example, that is called cross resistance. And then we have the co-resistance system, where the resistances to different antibiotics and metals are physically co-located on the same genetic element, for example on a plasmid. So co-regulation and co-expression are different terminologies, where expression of resistance systems to metals and antibiotics are controlled by a common regulator. So these are the different mechanisms of cross-resistance, co-resistance, and co-regulation, co-expression of metal and antibiotic resistance. So that is why the question was ability to resist a toxin because of genes encoded in the same plasmid as antibiotic resistance gene. So this is called co-resistance and not cross-resistance. Okay, so that is why the answer will be co-resistant. So we'll stop here today. I'm sorry, there's a lot of noise coming in the background, which is why I had to shift places. I'm really sorry. So I live in a hostel, so there's some kind of an event going on, so that is why there's a lot of noise. It's not really under my control. So I'll upload the video on YouTube, and if you have missed any portion of it due to the noise or anything, I'll try to reduce the background noise and upload it so that it's easier for you to see it. Otherwise, all these slides I'll upload somewhere, and the links will be shared to you, so you don't have to worry about that. So any questions related to what we discussed today? If you don't have any questions, maybe we can conclude the session and we can meet next day, same time, same link. Otherwise, we can discuss if you have any questions today. Also, Neha, I told you in the session also, I talked to the NPTEL staff, and they have said that they have actually forwarded my message, my concern to the, concerned faculty, concerned people at IIT Roorkee. So they are handling the NPTEL website and all the assignments they are only making. So there's some kind of technical glitch, I'm guessing. So they'll try to rectify it. So you won't have to suffer because of the technical glitch on the website. So all the correct answers will be checked, and they'll probably look into it. So you don't have to worry.
Okay, thank you, ma'am.
No problem. I also saw the discussion forum that you have actually posted 2 times, but there was no response. So I actually have no role in this because we don't handle the website. So but at least I could talk to the staff, and they have said that they will look into it. So hopefully this will get rectified, and You won't have face any
Okay.
problem from the further assignments.
Okay. Okay.
So anybody else has a question, or maybe we can. Really sorry about the noise thing, because we live in a hostel, so it's very difficult to get a place with quiet. And I was actually walking from my room, but it's very close to the uh, event where the event is happening, so that's why huge noise is coming from here. So right now I'm not in my room, as you can already see in the background. So really sorry for that. I'll try to reduce the background noise and upload on YouTube, so it will be easier for you guys to understand what I'm saying. So if nobody has any more question, we can conclude this session and we can join next time, next day, next Friday, same time, same link. Thank you, everyone.